In this video, we will discuss about pancreatic cancer glycosylation and the current treatment approaches. So how this glycosylation changes in the pancreatic cancer, that we will cover, and what are the current treatment approaches, the basic idea behind the current pancreatic cancer patients treatment approaches, that we will cover. So in the case of pancreatic cancer, there is a change in the glycosylation profiles that has been observed, and the major changes that people have seen is about this truncated O glycan, where we can see that the O glycans are truncated and there is only 2 or 3, that is TN or STN antigens are present. And in terms of the N glycans, they can see the highly branched N glycans that are shown here, and also this increased silations, that we can see that the salic acids are increased over these glycan structures, and this core and outer arm fucosylations. The fucosylations can be seen in the outer arm of this glycan structures, and these things are not commonly found in the normal pancreatic cells. And about the treatment approaches, so first one, the treatment approaches is about using this drug, this glycosylation. What it will does is it will attach, or it will compete with this HMGB1 and S100, and this is commonly known as receptor for advanced glycosylation end product. It will bind to this HMGB1 and S100, and thereby preventing the intercellular signalings that might cause invasion, metastasis, and even resistance to drugs such as 5-FU and gemcitabine. So these things can be inhibited just by targeting the receptors for advanced glycations and products. And the other approach is use these drugs, that is mineralide, and this will get cleaved in this liver using the enzyme called this esterases, and thereby convert it into the active form, that is triplotide. And this triplotide has a role towards suppression of this XPP, and this XPP suppressions thereby cause or prevent these c-MYC expressions in the cytosols, which is mainly expressed when the mitochondria is not working properly. And once its expression is suppressed, then it will prevent this tumor proliferations, and thereby the major causes of the pancreatic cancer is its aggressive migration and proliferation properties. If that is suppressed, then it's easy to diagnose the patients with such conditions. The other things that is commonly used is about the related to this CAR T cells, where through this leukapheresis the T cells are being extracted from the patients, and thereby they are genetically modified to target this KRAS G12D T cell receptors. And once they are genetically modified, they are transfused again back into the patients, and thereby it will help this, since T cells already has the receptors to target the pancreatic cancer cells, it will help in the suppression of the tumor proliferations and immune evasions. These are the 3 main approaches that are mentioned in this paper. And about the change in the glycosylation profiles that are usually observed in the pancreatic patients who are affected with the pancreatic cancer. And these glycosylation profiles can also be formed as one of the biomarkers. And one of the biomarkers, for example, is CA19-9, which is the biomarkers based on this glycosylation profile. However, it's difficult to distinguish whether this CA19-9 is coming from the normal pancreatic cancer cells or healthy or cancerous normal pancreatic cells or cancerous pancreatic cells, because some nominal level of CA19-9 are also present in the normal pancreas. So this is one of the reasons that CA19-9 does not gain so much attractions towards further research, but some combinations of these carbohydrate structures can be used as a biomarkers. That is, if those combinations are present, then this could be the indication of the pancreatic cancers initiations within the patients, as usually these pancreatic cancers are difficult to detect at the early stage, and having some biomarkers will definitely give us a boost towards diagnosing these patients who are affected with this pancreatic cancers at the early stage. So that's all for this video. If you need to understand more about their topics in detail, you can go through the reference that has been provided in the description. Thank you for your time.